Applying a stimulus to the cell membrane can change its electrical potential. If the stimulus has enough energy, it generates an action potential. The action potential propagates along the neural membrane away from the stimulated point. Voltage-gated sodium channels open when the neural membrane voltage becomes positive enough. Sodium ions flow into the cell and depolarize the cell membrane. Depolarization propagates along the entire length of the nerve fiber by opening the voltage-gated sodium channels located throughout this membrane and the influx of sodium ions. The action potentials or the nerve impulses flow forward and cannot be regenerated in the areas of the neural membrane that are in the absolute refractory phase. The method and speed of impulse propagation in myelinated nerve fibers are different from unmyelinated nerve fibers. Myelin is a multi-layered membrane that wraps around the axon. The spaces of the axon that do not have myelin sheath are called the nodes of Ranvier. These nodes are 2 to 3 micrometers long and are spaced about 1 to 3 millimeters apart. In the peripheral nervous system, the myelin sheath is made by Schwann cells, and in the central nervous system, it is made by oligodendrocytes. In myelinated fibers, the action potential is generated only in the nodes of Ranvier and jumps from one node to another. This impulse propagation procedure is called saltatory conduction. The myelin sheath prevents ions exchanges across the neural membrane. Sodium ions enter the cell through the voltage-gated sodium channels located in nodal areas. The influx of positive ions across one channel and following that, the flow of positive ions inside the axon stimulates the next channel and depolarizes the cell membrane at that point. Saltatory conduction has two important benefits. Saltatory conduction increases the rate of electrical signals transmission in myelinated fibers. Also, more metabolic energy is saved because the ions exchanges take place only in nodal areas. In unmyelinated fibers, sodium channels are located throughout the axonal membrane, and depolarization occurs in all parts of this membrane. Therefore, the impulse propagation rate decreases while the energy consumption increases.